Hey guys, my name is Emily Landrum, and my partner for this assignment is Emily Thiery. I've been working really hard to bring you guys this presentation on embryonic vascular development, and due to current circumstances, this presentation will be recorded in two parts, both of which will be available on YouTube. We're also going to upload a link to the PowerPoint we made so you have full access to what you need to study on the final exam. But with that, let's get started. Maybe. There we go. So we've learned a lot of information over these last few months, and I really hope that by this point you can appreciate just how complex the molecular and physiological processes are in embryonic development overall. Knowing this, it really shouldn't surprise you that there are several steps that need to happen in order to establish a fully functioning vascular network from the origins of fertilization. Fortunately, we don't have to cover the first weeks of development past the point of fertilization because we're pretty well acquainted by now, I would hope. So we're going to start here, or at around the first stages of somatogenesis when you see those first few pairs within the embryo. Vascular development will then proceed from this point onward and throughout life after birth through a series of processes that ultimately work for the purpose of circulating oxygen, nutrients, and molecular signals to and from various tissues throughout the body. So this is a really cool study that we found to sort of represent the different stages of vasculogenesis. And it was done using mouse embryos, and they were able to map out the vasculature at progressive stages, of somatogenesis as indicated by these little numbers here. These are actually the increasing numbers of somite pairs. So A is 5 to 6, B 11 to 12, etc., etc. And they end up giving this final product as seen to the left, which is the developed vascular network, I think by the end of body folding and everything after that was established. So these first images here, A and B, actually are outlining the first stages of vascular development, which includes the de novo establishment of primitive vessels. And this is through a process called vasculogenesis. As you can see, it produces a discontinuous network, there we go, of blood vessels that are starting at multiple different points throughout the embryo. And these are going to have to follow different signals from hypoxic tissues and surrounding or neighboring vessels so that they can merge into each other and become a functional continuous vascular network. But it's still not the mature or the complete vascular network. And so it's actually called the primary capillary plexus in the second image here. Further development will be induced by a process called angiogenesis or vascular remodeling. So this is the remodeling of the primary capillary plexus, as well as the continued growth and development of those vessels that are currently in place. And those can be seen in these two bottom images. And they sort of have these vessels outlined, and you can sort of see that they're becoming thinner. The complex is way more difficult or way more complicated, and it's pretty cool when you get to that left-hand image over here. And I hope you guys can really appreciate how awesome the network is and that it follows the components of other systems, such as the central and peripheral nervous tissues. And it's really important to realize that both of the stages of vasculogenesis and angiogenesis involve cell differentiation and migration to take place before they can even start. So that's really where we're going to begin, and then we'll move forward from there. Awesome. So like we just mentioned, vascular development is going to start around week 3 after fertilization with those first few somite pairs. And it's going to begin with the differentiation of cells within the mesoderm into specialized cells called hemangioblasts. And this is going to be done by the process of epithelial to mesenchymal transformation. Like other specialized cells we've learned about in this course, these hemangioblasts are actually going to migrate throughout the mesoderm into the yolk sac as the body is preparing for folding. In the yolk sac, these hemangioblasts are going to start to aggregate with one another, and they're going to form these clusters called blood islands, which are actually visible within the layers of the mesoderm and the endoderm of the yolk sac at around week 4 after fertilization. Cool. So within these blood islands, the hemangioblasts are actually going to differentiate into 2 distinct cell types. The first cell type that we're going to cover are endothelial precursor cells, and these are going to be the cells that line the periphery of these blood islands, and they are going to contribute to, I guess, the vessel walls. And so we actually are going to refer to them as the bricks of our blood vessels. And they're going to be the primary cells that are influenced by the processes of vasculogenesis and angiogenesis. On the other hand, cells that are located, or hemangioblasts that are located centrally within these blood islands, are going to differentiate into what is known as hematopoietic stem cells. These hematopoietic stem cells are pretty cool, and some of you may have heard of them before, as they maintain a multipotent nature. And that's not limited to embryonic development alone, but it's actually conserved throughout life and occurs within the bone marrow of all of our bodies, as these cells will contribute to all of the different components of our blood, as well as they will contribute to some of the organs of the embryo through a process called hematopoiesis. We're going to talk about both of these cell types in a little bit more detail, but we're going to start out with the hematopoietic stem cells. So earlier in the semester, we learned that the yolk sac is the primary location of hematopoiesis in early embryonic development. Hint, hint, remember that. Now you know why. Hematopoietic stem cells differentiate into specialized cells by way of hematopoiesis. Hint, hint, we've said it like 3 times now. Maybe you should know that. There are going to be 3 categories of cells that are derived from these hematopoietic stem cells through this process of hematopoiesis. Those cell types are myeloid, lymphoid, and progenitor cells. The myeloid cells are actually going to be the components of your blood, or the majority of the components in your blood, including your erythrocytes, your platelets, and your innate immune cells, like your neutrophils, your macrophages, monocytes, stuff like that. Your lymphoid cells are actually going to be cells of your adaptive immunity, or your B cells and your T cells. And the hematopoietic stem cells that actually contribute to your lymphoid cells have a different point of origin than your myeloid and progenitor cells. We'll talk about that in a minute, though. Lastly, your progenitor cells are going to include everything else. So it's kind of like the junk drawer of hematopoietic stem cell derivatives. But they're pretty versatile and kind of cool that they give rise to myeloid cells, or they can contribute to that development of the various organs that we're going to talk about in just a minute. But before we move on to the migration portion of these hematopoietic stem cells, we're going to ask our first question. These questions are going to be asked throughout the presentation, and they're for your benefit since they're probably going to be on the exam. So make sure that you pay attention and, give your best effort to participate from home as much as you can through this virtual process. So question 1, hematopoietic stem cells are first seen. And I'm not going to read those out loud, but I'll give you a minute to answer the question. Yep, that's enough time. If you answered C, yolk sac, you would be correct. See, hematopoietic stem cells originate within the yolk sac, as it's the primary location of hematopoiesis in early embryonic development. However, we all know that the yolk sac is going to eventually degrade, meaning that yes, some type of migratory event must take place for the process to be maintained throughout development and onward, which we can see is actually the case, because there are 2 waves of hematopoietic stem cell migration, and they originate from different locations. The first one is going to be originating from the yolk sac, or that primitive area, or that area of primitive hematopoiesis. And these cells are going to become the myeloid and progenitor cells, and they're going to migrate to colonize the primordial liver. The second wave of migration is going to originate from the AGM, or the aorta gonatomesenteris. And this area is going to include 3 different structures, including the dorsal aorta, the genital ridges, and the mesenterus, hence the name aorta gonatomesenteris, or AGM. And these are pretty cool in the sense that they are going to contribute to the process of definitive hematopoiesis. And these derivatives from the HSCs of this process are going to give rise to the myeloid, lymphoid, and progenitor cells. So all 3 cell types, or all 3 cell derivatives.